All right, so picking up where we left off, solutions that have a higher concentration of the solute than the cell are said to be hypertonic. Hyper meaning greater than, and tonicity again referring to the strength of the solution. If a cell is placed in a hypertonic solution, water is going to leave the cell. So water is going to rush out of the cell, and the net movement of water is from the inside to the outside of the cell. Any solution with a greater concentration of 0.9% NaCl is hypertonic to red blood cells. Animal cells placed in such a solution will shrivel due to water loss. This process is referred to as crenation in red blood cells. Plant cells placed in a hypertonic solution will experience plasmolysis, or the shrinking of that cytoplasm away from the cell wall. So the plasma membrane is going to prevent the passage of most substances, but biologically useful molecules are able to rapidly enter and exit the cell by either way of channel proteins or because of carrier proteins in the membrane. These transport proteins are specific, so they can only transport a certain type of molecule or ion across the plasma membrane. The direction of the transport is going to follow the concentration gradient, moving from an area of high concentration to an area of low concentration. So no energy is going to be required for the process of facilitated transport. So in the event that we have a lot of, let's say, water molecules on this side of the plasma membrane and very few water molecules on this side, water is going to move from an area of high concentration to an area of low concentration, but it's going to require the use of either a channel protein or a carrier protein to help get it across. So here's our bit on facilitated transport. It's just the movement of molecules across the plasma membrane from an area of higher concentration to the area of lower concentration via a protein carrier. This is not going to require any energy, and these protein transporters are going to be highly specific. They're only going to transport one particular type of molecule. Active transport. Active transport is another method for getting molecules across the plasma membrane, but in this case, we're going to require some energy. There are times when a cell might need to increase the concentration gradient across a membrane in order to accomplish more work. In other words, we are going to be moving up or against the stream. So it's kind of like if you were rowing in a rowboat down a river. If you're moving with the current, so you're moving down the concentration gradient. If you're moving from upstream to downstream, you can kind of just sit in your boat and not expend any energy and just sort of float downstream. In the case of active transport, we are going to be moving against the concentration gradient. We're going to be moving against the tide. We're moving from an area of low concentration to an area of high concentration. Just like rowing upriver, it requires some energy. Going against that concentration gradient is also going to require some energy. So this process of moving molecules against the concentration gradient is called active transport. We're going to be moving from regions of low concentration to regions of high concentration. This transportation is also going to require the aid of a carrier protein to move those molecules across the membrane. Active transport is also going to require the input of energy, typically in the form of ATP. This is going to help bind the substrate to the carrier protein and carry it across the membrane. Proteins involved in active transport are often going to be called pumps. This is because of their similarity to water pumps that require energy to push water against the flow and force of gravity. We're going to use these pumps to push molecules against the force and flow of their concentration gradient. One type of pump that is active in all animal cells, but is especially associated with nerve and conduction cells, would be the sodium ion pump. It's going to move sodium ions to the outside of the cell and potassium ions to the inside of the cell. The transport of the sodium and potassium are linked together through the same carrier protein called the sodium-potassium pump. It's going to carry these molecules by undergoing conformational changes to bind to these two ions. So I'll show you in the next slide. Here we have our sodium-potassium carrier protein. It's going to have an initial shape that is designed to allow potassium ions to bind to it. So we're going to bind, not potassium, sodium ions. We're going to bind 3 sodium ions to it. A phosphate group is then going to be broken off. We're going to break our ATP and release a phosphate group that can then bind to our sodium-potassium pump. Once we bind to the pump is actually going to undergo a conformational change. Instead of being open on this end, it's going to close on this end and open on this side. It's going to force these sodium ions out of the cell and into the sort of interstitial space, or the area outside of the cell. The potassium from the original ATP molecule is going to leave. Once it leaves, it is removed from, once it opens on this side, we're going to allow potassium ions to come and bind into place. Once they have bound into place, the phosphate group is going to be released. When it releases, we're going to undergo another conformational change. The potassium sodium pump is going to close, and it's going to release its potassium cargo on this side. Once it's released on this side, we're now going to allow more sodium ions to come in, bind in. Once they have locked into place, ATP is going to be broken. We're going to undergo a conformational change. We're going to release our sodium ions, and we're going to pick up more potassium cargo. Once the potassium is in place, this phosphate group is going to leave. We're going to snap shut. We're going to then release our potassium on this side of the plasma membrane, pick up some more sodium, and the cycle is going to continue. All of this process is going to require the use of ATP, and so this is how we require energy, or we use energy in active transport. It's always going to require ATP and the use of a carrier molecule to move those molecules against their concentration gradient. Bulk transport is another method of active transport. Some molecules are going to be transported across the membrane via protein channels and carriers. However, large macromolecules, things like whole proteins, polysaccharides, nucleic acids, they're going to be far too large to be transported this way. So they're going to have to be transported through vesicles. In this case, macromolecules are going to exit the cell through exocytosis and enter the cell through endocytosis. And we're going to look at some of those here. Endocytosis occurs when the cell takes a substance by forming a vesicle around the material. So here we have a material that the cell wants to ingest. We're going to actually form a vesicle around it and then pinch off and bring that vesicle inside. Once that portion of the membrane wraps around the substance, it's taken in, pinched off to form a vesicle. That vesicle can then be filled with digestive enzymes that can break down those substances and used by the cell. There are three forms of endocytosis. There's phagocytosis, where we're going to be taking a large solid material in through endocytosis. Penocytosis is also referred to as sort of cellular drinking. This is where we're going to form a vesicle around a liquid or very small particles. We're going to just kind of sip tiny little particles, and we're going to bring it in these smaller vesicles. So that's our penocytosis. And then there's also receptor-mediated endocytosis. Receptor-mediated endocytosis is a very special form of penocytosis. It's going to require receptor proteins that recognize materials to be brought into the cells. It's going to specifically recognize those particles and bring them in individually. Molecules like vitamins, peptide hormones, lipoproteins can bind to specific receptors found in special locations in the plasma membrane called a coated pit. So this coated pit has these specific receptor proteins that are going to bind to their cargo. Once they have bound to their cargo, they're going to pinch off and bring those molecules inside. This process of receptor-mediated endocytosis is highly selective and very efficient. Exocytosis is going to be very similar to endocytosis, but in this case, we are going to actually be removing materials. We're going to be releasing them from the cell. So exocytosis transports molecules outside of outside of the cell via the fusion of a vesicle with a plasma membrane. So it's kind of like a reverse endocytosis. We're going to have our little cargo. We're going to bring it up to the plasma membrane. We're going to fuse our vesicle with the plasma membrane and actually release that material to the outside. These vesicles are going to come from the Golgi body. And we're going to talk about the Golgi body in a few minutes when we talk about the different organelles to the cell. We're going to see a lot of things. So it's not just waste that is going to be excreted from the cell in this manner. Also things like hormones and digestive enzymes, neurotransmitters are all going to be excreted or secreted from the cell like this, where they're going to be brought to the surface and then released to the outside. All right, so you should be able to summarize how the fluid mosaic model describes the structure of the plasma membrane, compare and contrast diffusion, osmosis, facilitated transport, and active transport, and discuss the various ways that cells can move materials in bulk both into and out of the cell.